Here he is, broken boy, with the bad leg. The reason why I'm not in today. He's doing okay though, aren't you, buddy? Say hello. Anyway, I'm going to get him out of the way because he's not super comfortable there. So today's lesson is about large-scale collaborative genetic projects. Syllabus point 552, last syllabus point in this topic. As you can see in the syllabus dot point, it's all about collaboration, which I'm sure you guys are familiar with, but that's what we're going to be starting with, talking a little bit about collaboration. Now, in terms of notes for this section, you don't necessarily have to take notes for this, but maybe just have a think along the way whether or not you need to. It'll become a bit more apparent at the end. Let's have a look at the syllabus dot point. Investigate the use of data analysis from large-scale collaborative projects to identify trends, patterns, and relationships. For example, and I've highlighted here the use of population genetics data in conservation management because that's the one I'm going to focus on today. The reason why I'm focusing on that one is because you already did it in your assignment, hopefully, if you did your assignment right. So you might already know most of what I'm going to talk about, and therefore you won't need to write anything down. But it is important to note that there's the two other options here as well, looking at the inheritance of diseases or population genetics in human evolution. We'll look at them a little bit more later in the course, but if that's something you're really interested in, you can by all means go and research that as well. Those are just 3 suggestions, so we don't have to do all of them, and given how much content's already in the course, we're not going to. We're just going to focus on the top one up there. So for today, we're going to be looking at conservation management, but like I say, the others are genuine options that you can do, and you'll learn a lot more about the disease one in the next topic that we do. So very briefly, I know you guys are probably already familiar with collaboration, but we better run over really quickly what collaboration is. Basically, collaboration is working together, whether it's for business benefit, academic benefit, the greater good, whatever it happens to be. Collaboration is what happens when groups with different interests and different skill sets get together and work together towards a bigger project or a bigger idea. And basically the whole advantage of collaborating is that you get all these different skill sets and all these different bits of information, and you can put them all together in one place and hopefully produce something that is better and more detailed than what any one of those people could have produced by themselves. Right, so to set the scene a little bit, we need to talk a bit about human impacts on the environment. So basically, we're all very familiar with that story, or that narrative that we live in, that humans are basically destroying the world. We impact on almost every part of the world. We've killed about, or I shouldn't say we've killed, we have driven the extinction of about 83% of all wild animals and about half of all plants on Earth. And right now we impact on roughly 75% of the Earth's surface, and 60% of our world's large mammals are threatened with extinction. Now, 75% of the Earth's land surface might sound like a fair bit, but not that much. But if you have a look at the map of where human impacts are, this is the map on the screen now. I'm just going to use my cursor to point here. On land, you can see that low impact is indicated by sort of these lighter colors. And basically the only areas that you're going to find low impact are right up the top of the North Pole here and right down the bottom in Antarctica. And basically all of the continental land masses outside of those are heavily impacted on by humans. So you got to remember that 75% actually means something like 99% of habitable zones on Earth. Now, we probably already know all the ways humans impact, but here's a few examples I just thought I'd put in to give you some perspective. So we've got pollution. Here we've got improper water management, so things like the way that we irrigate the Murray River in Australia, for example, can lead to salination of some parts where it gets too salty, and it can just completely use up all the water in other parts. And you can see in this picture here we've got a dried out riverbed, and here we've got deforestation possibly caused by mining or development or farming or something like that. Just one of many different examples. So when it comes to conservation, historically we haven't done particularly well in terms of the outcomes that we have achieved for plants and animals on Earth. There's a few reasons for that, and a few of them are listed on the screen here. So we tend to have a territorial approach to conservation, which means that we like to protect the parts that we have ownership over, and we like to dust our hands clean of the parts that we don't own or control, because it doesn't really matter to us politically and territorially. We're not responsible. We've always got internal conflicts. A really good example of that right now, if you have a look at the Liberal government and the funding of the Adani coal mine, for example, you'll find that within politics in Australia, you don't have everybody agreeing on what should be done for the environment, and as a result, we often have these large-scale destructive processes approved for economic reasons or because there wasn't enough people to argue against it. There's always competition for funding. It's not always the case that the best company gets all of the money. If you want a classic example of that, have a look at the gigantic big burst of funding that the Great Barrier Reef has just been given by a relatively unknown charity group. You can Google them and have a look at that. There's been really big controversy. Basically, the government just opted to give them a bunch of money without any sort of competition, which is quite unusual. Normally there's always competition for funding, but the key point here is that not everyone gets the money, right? And so that means that not everyone gets an equal chance at performing some sort of conservation service. Influence and recognition, again, sort of ties into that other idea. Basically, what we're saying here is that if you have the means to draw attention to yourself with money or fame or internet stardom or whatever it is, then you have a better chance of getting funding for your project. And field projects tend to be isolated. In other words, people historically have worked independently of each other, and as a result of that, they haven't shared their visions very clearly and haven't linked them. Classic example from our local area here is the Bushrangers Bay Aquatic Reserve. If I just go back to that other slide, what we said was that field projects tend to be isolated. If you have a look at the Bushrangers Bay Marine Reserve at Bass Point in Shell Harbour, this little dotted area here is the Bushrangers Bay Aquatic Reserve. And I'm not sure if you can see down the bottom here, but this little scale bar down the bottom only represents 500 meters. So this is less than 500 meters long and less than about 100 meters wide at its widest point. This is a tiny marine reserve. Fantastic for people who want to come and go for a snorkel and things like that, but in terms of actual conservation, very ineffective. It's way too small to accomplish anything. It's isolated, and that was the point we were talking about on the previous slide. Okay, so conservation genetics. This is going to be the backbone behind the big projects that we're going to talk about. And as the name would imply, when you take conservation biology and you take genetics and you put them together, you make conservation genetics. Basically, when we are damaging the environment, we are harming animals and plants and organisms, and so therefore we need to effectively manage them to make sure that we can preserve threatened and endangered species. That's where conservation biology comes in. Basically, conservation biology is all about looking at populations of organisms, how they've been affected by various factors, be it people or environment change or weather or whatever, and learn a little bit about them. So that's conservation biology. And if you tee that up with genetics, which is looking at inherited characteristics, which we're all familiar with, and put them together, you get conservation genetics. In other words, taking all of that information, we can learn about what has happened to populations, and then sequencing out the genetics and seeing what we can learn from the genetics of those organisms, put that together, conservation genetics. And that gives us a really powerful tool for investigating how genetics can be harnessed to give organisms and populations a better chance of survival. There's just a little picture here that I thought I'd put in. It's just showing how we can map out the genes of different organisms, much like you guys already did in your assignments. These colored bars, you can probably imagine, represent different amino acid sequences, and you can see we have closely related species, and a conservation geneticist would look at these and look at patterns in the populations and how they've changed over time, and hopefully put together a little bit of a picture as to whether or not genetics is influencing their survival. Okay, so I'm going to talk about some collaborative projects now. Remember that this is the key part of the syllabus. This is sort of the nuts and bolts of what we're talking about. We need to know about large-scale collaborative projects that are dedicated to conservation. And before I talk about this one, I want to remind you, if you think back and you think, I already did that in my assignment, then you don't need to know these other ones, because you just need one good example. And if you've already got one, there's no point memorizing 2 or 3 more. You're not going to be required to know multiple examples. But I'd recommend you listen through these and make sure that yours is going to meet the requirements for the syllabus. And I've got a little guide at the end that you can use to make sure. So the first project I'm going to talk about is to do with the Tasmanian devils. If you have a look up at the top right here, you can see a picture of a Tasmanian devil that is suffering from devil facial tumour disease, which is a really big problem in Tasmania. Basically, the Tasmanian devil population there has a contagious form of cancer, which is a very rare thing in nature for cancer to actually be contagious. They pass it to each other. And it's really become a huge problem in very recent history. In the last decade or so, it's become a much bigger problem than it had been before. Basically, the way that this works is that the cancer cells get spread by biting. So because the devils have infected faces, infected with tumors, and when they fight, they bite each other, they have a tendency to scratch those tumors on their faces and rip them open and transfer bits of blood and tissue into each other's mouths. And in doing that, the cancer actually gets passed on. So very uncommon in nature, but it's happening here, and it's a big problem in Tasmania. Here's a picture up close. I know it's very sad, but it's important that we understand what we're dealing with here. And I want to draw your attention up the top here. I've put these couple of groups up here. There's actually more that are involved in this project, but these are kind of the key players. So I'm going to be talking about the University of Tasmania, the Tasmanian Government, the Forest Practices Authority of Tasmania, and Save the Tasmanian Devil program. In terms of what you need to know, you don't need to remember all of those. But it's important that you understand that we're talking about multiple large groups here. So each one of these groups or organizations could have, anything from 1 to 100 people working on this particular problem. Okay, so the goal of this program is to bring together genetically diverse individuals into a secure managed area and breed them to create a managed population in case the species goes extinct. Now, if we have a think about what we're saying here, first of all, you'll notice bring together genetically diverse individuals. We're talking about genetics. It's really important that we understand that this conservation project, a project whose goal is to conserve the Tasmanian devil, is using genetic data to do so. Now, getting genetic data is not something that a lot of government bodies would have access to. They need to use laboratories. So they're going to be contacting the University of Tasmania to use their facilities and their expertise in order to get that information. Next thing to look at is the conservation side of things. So that was the genetic side, the conservation side, trying to create this secure managed area where the disease does not exist. Okay, a sanctuary. That's a very classic conservation strategy. And at the same time, breeding them and keeping them kind of as this little secured population, so that if for whatever reason the wild population gets totally decimated by this disease, there is another population they can use to repopulate that is a healthy population. So we've got a genetic component and we have a conservation component. In terms of collaboration, we see that we have genetic research being conducted by the University of Tasmania, and that data is then used to strategically zone populations by the government. So there's a very obvious piece of collaboration. The science is done by the scientists, the zoning is done by the politicians, and the two are communicating together. Very important to note that this isn't something that traditionally has happened. For a very long time, there's been very little collaboration in science, so this is a relatively new approach. And then that greater data, so the combination of the University of Tasmania and the Tasmanian government and their zoning, then goes back to the Save the Devil program. They're doing things like raising money, running breeding programs, employing volunteers to go out and do surveys, all those extra things that are going to boost and help out the University of Tasmania and the Tasmanian government. And overseeing all of that will be the Forest Practice Authority. They're kind of like an environmental protection agency, and they'll make sure that anything that the government decides to do or that the university decides to do is not going to do further damage to the environment. Okay, I've got a little video here. I'm going to try and play it. If this doesn't work, I'll edit this part out and then I'll insert the video in a moment.
The Tasmanian Devil program began in 2003, and it was started as a collaboration between the Tasmanian state government and the Australian federal government in response to devil facial tumour disease, or what we call DFTD. It's a contagious cancer that was sweeping through the Tasmanian devil population throughout Tasmania, and the program was set up for us to work out exactly what the cancer was, what effect it was having on devil populations in the wild, and what we could do to help those populations. I've been with the Devil program for 10 years, and from the first year that I started with the program, I've been wanting to come to the southwest of Tasmania to have a look at devil populations here. The southwest of Tasmania is considered probably the most remote part of our state. It's very difficult to get to. The only way you can get here is either by boat or by helicopter. So we've been talking about this trip for a very long time. We spend quite a lot of time working in the field with wild devil populations, understanding the effect of disease on these populations. But the populations down here in the remote southwest, we don't know how big the populations are. We don't know whether they're diseased or whether they're healthy. We suspect that they are genetically more diverse than devils over the rest of Tasmania. But we don't know any of this because nobody has ever trapped these animals before. Has never been and had a look at these populations before. So there's always been a bit of mystery about the devil populations here. We're looking at making the most of the genetics that we have in our insurance population and boosting those, but also making sure that the animals that we're putting back into the wild have the greatest genetic diversity that we can give them. So this trip now is the beginning of what we call the Founder Project, and the Founder Project is about understanding how we can maximise the genetic diversity in our insurance population and enable that genetic diversity to be passed on into the wild populations that we're supplementing with our translocation program. I guess it's just a far more complicated trip to organise. It's a bit more complicated when you're here, but it gives great rewards, or at least we hope it does at the end.
Alright, so you can see in that video, number one, talking about the genetics and typical conservation strategies. I don't know if at the end you noticed, but they had all of the logos of the partnerships that they've made to get that program to happen. And obviously looking at the genes of those populations and talking about creating a genetically strong and diverse population in the wild remotely that can potentially repopulate the rest of the population if it needs to. Alright, I'm going to talk about a second example now, the Universal Protein Resource, or UniProt, which hopefully sounds familiar to everyone because you'll use this in your assignment. And yes, this is another example of a tool that represents collaboration, large-scale collaboration between scientists around the world working towards conservation biology. So, first of all, the project itself is set up as a collaboration. So we had the European Bioinformatics Institute, the SIB Swiss Institute of Bioinformatics, and the Protein Information Resource all came together to build UniProt. And more than 100 people work at any one time on UniProt, doing things like curating the database, software development, support, building the website, all that sort of stuff. So the reason why I wanted to include UniProt is I think that it kind of represents the future of scientific collaboration, because you've got collaboration on demand. What I mean by that is that the data is available to be accessed anytime on your own terms, however you like. And so you can take data, share data, post data at your own accord. You can use it to collaborate without borders. This is a totally online and digital tool. Biologists all around the world can use it. They can change the language of it. They can get the information they need. They can share the information that's important to them without having to worry about being in contact with specific scientists. It's freely available. I think that's really important because going forward, having these large-scale free tools are going to create a whole new economy of collaborating in science. And theoretically, it could change conservation forever because you reduce or you minimize so many of the impacts that many countries run into when they try to run scientific conservation projects. If they don't have a very big budget, if they don't have many scientists working in the country, if they don't have access to the tools you need to do genetic sequencing, they can do all of this online using pre-existing data. So what I did was I ran a little search on the UniProt website. I'm just going to pause that and I'll run that again in a sec. So you saw it coming up there a second ago. It's going to come up again in a moment. What I did was I went onto UniProt and I searched for scientific papers that have used UniProt in their references. In other words, scientific papers that quote UniProt and also quote the word conservation. In other words, papers that used papers that were themed around conservation that made use of the UniProt website. This is what I got. So you can see it coming up now. So every one of those blue headings is a scientific paper. You can see the authors are attached there, the years are attached there, all that sort of stuff. It keeps going. Unfortunately, my animation stopped there, but down the bottom there was actually 2,500 more papers. So it has been used significantly. Right, let's talk about meeting the requirements of the syllabus for this dot point. So, you may already meet the syllabus with the work you did in your assignment. You may not. Depends what you put in your discussion. These are the key points I think you need to think about. If you can't meet them, you need to go back in this video and take some notes or do some research of your own and take some notes. These are the things you need to know. Can you name an animal with which scientists are collaborating to share genetic information with an aim around conservation? Number 2, can you talk about the groups that are doing that and talk about how they work together? Number 3, can you discuss the importance of collaboration in general? Why it's important for scientists to collaborate? And number 4, can you justify why money should be spent on collaborative projects that focused on endangered populations? The reason why I put that last one on there is I think that there's a chance that that could be a HSC question in the future. You know, an extended response question where they'd basically say something like, justify the large expenditure on genetic research to collaborate across borders to protect endangered species. And what you need to be able to talk about there is why conservation and the future of conservation is all about collaborating. It's all about incorporating genetics into your strategies for conservation, and it's all about I guess working smarter, not harder when it comes to conservation, because we haven't been very successful in the past. So have a look at those dot points that are up there on the screen. Make sure you can tick off each one in your head. If you can't, like I said, go back and take some notes.